Welcome to our channels. Last class we will discuss about in general microbiology and unit third isolation of algae and methods of cultivation of algae and algae distributions and classifications and economic importance and so on. So discussed about it. So now we discussed about it strain selection and large scale cultivation of algae. Introduction: algae are used in food, feed, pharmaceuticals, biofertilizers, pigments, and biofuels. See, the success of cultivation depends on choosing the right algal strain and using suitable large-scale culture systems. First one, the strain selections. How is it strain selection? What are the criteria is followed? Selecting strains. First one, high growth rate must multiply rapidly under the culture conditions. And second one, high biomass. So product yield. It should accumulate proteins, lipids, carbohydrates, or pigments. And third one, environmental tolerance. Withstand changes in temperature, salinity, pH, and sunlight. And low contamination risk. Fourth one, it naturally resistant to contaminations. For example, spirulina grows in alkaline medium where few competitors survive. And fifth one, easy to harvest. Larger cell size or colony fermentations simplifies harvesting. And genetic stability, stable traits without frequent mutations. And seventh one, specific product requirements. So spirulina, protein, vitamins, and chlorella, single cell protein and biofuel, and tonalella, beta carotene, and diatoms, oils, and silica, anapina, nostoc, and biofertilizers. This is the specific product requirements also. And third one, large scale cultivation of algae. It's the open culture system. First to maintain for the open culture systems. And the types natural bonds, raceway bonds, circular bonds also. And features shallow bonds 20 and 30 centimeter depth. And pedal wheels maintain circulations. And sunlight as energy sources. Nutrients are added. And what are the advantages? Low cost and easy to maintain. And disadvantages: contaminations and environmental fluctuations and low productivities also. And for example, open culture system, for example, spirulina and chlorella cultivation in India, Mexico, China also. And next, B is a closed circular systems. This is the photobioreactor. And transparent tubes, panels, or tanks, it supply with sterile nutrients, CO2, and artificial light. And types tubular, flat panel, vertical columns also. And advantages: closed culture systems, controlled environment, pH, temperature, CO2, light, and high biomass productivity and low contamination risk. And disadvantages: it expensive to construct and operate. Examples used for high value product like beta carotene, dunalella, and hematococcus. And next, see hybrid systems, combinations of photobioreactor. This is the open bonds. For example, inoculum developed in BB are transferred to open bonds for bulk growth. And fourth, factors affecting large scale cultivations. Light, first one is a light intensity and photo period 16 to 8 hours, and temperature maintained 20 to 30 degrees centigrade for most strains. pH it usually 7 to 9, and this is the alkaline for spirulina. And nutrients: nitrogen, phosphorus, and trace elements. And aerations, the CO2 it supply enhance the photosynthesis, and mixing it prevents settling and increases light exposures. And fifth one, applications is food and feed So algae bar, spirulina and chlorella is a single cell protein, and biofuels, microalgal lipids and biodiesels, and pharmaceuticals, the beta carotene and astaxanthin. And biofertilizers, blue green algae, anapina, nostoc, and wastewater treatment, algae absorb the nutrients and heavy metals. And conclusions, strain selection ensures high yield, product specificity, and resistant to contaminations. And second one, large scale cultivations can be done in open ponds. This is the low cost. Or closed photobioreactors are high productivity. And third one, all together, these methods support the use of algae in food security, green energy, and unsustainable industries also. Thank you. Please subscribe our channels. Thank you, viewers.